Good morning, everyone, and thank you for joining me today. We're going to dive into a comprehensive overview of forensic DNA analysis. This presentation will take us from the basic genetic fundamentals all the way through to the modern applications, challenges, and ethical considerations involved in this fascinating field. I'm excited to explore these topics with you and shed light on how DNA analysis plays a crucial role in forensic science. Let's get started. Forensic DNA analysis is a powerful tool that helps us identify individuals through their unique genetic information. This process plays a crucial role in legal investigations by providing concrete evidence that can link a suspect to a crime scene or victim. Beyond solving crimes, forensic DNA analysis is also instrumental in exonerating people who have been wrongly accused or convicted, ensuring that justice is served accurately. Its significance extends to law enforcement, where understanding and utilizing DNA evidence has become an essential part of modern investigative procedures, strengthening the overall effectiveness of the justice system. DNA is structured as a double helix, made up of four nucleotide bases: adenine, thymine, cytosine, and guanine. These bases pair up in a specific way, forming the rungs of the DNA ladder. Genetic inheritance is all about how this DNA, with its unique sequence, is passed down from parents to their children. While most of our DNA is similar, polymorphisms introduce small variations that make each of us unique. These genetic differences are crucial in forensic science because they allow experts to identify individuals based on their DNA, making a powerful tool in criminal investigations. When it comes to forensic DNA analysis, the sources of samples are quite varied. Commonly, we collect DNA from blood, hair, saliva, or even skin cells. Each of these sources can provide critical genetic information needed for investigations. However, it's not just about collecting the sample. Using proper collection techniques is absolutely vital. If the sample gets contaminated or damaged during collection, it could compromise the entire analysis. That's why careful handling is crucial from the moment the sample is collected. Once collected, preserving the sample properly is equally important. This means using controlled storage and careful handling to maintain the DNA's quality. If preservation isn't done correctly, it can lead to degradation, which then affects the accuracy of the lab testing. So, ensuring both proper collection and preservation methods are followed is key to obtaining reliable forensic DNA results. When we talk about laboratory methods in DNA analysis, the first step is DNA extraction. This process isolates genetic material from the samples we collected, which is crucial because we need pure DNA to work with. Next comes quantification, where we measure how much DNA we actually have. This helps us determine if there is enough material to proceed with further testing. Once we confirm the quantity, we use PCR amplification. This technique allows us to make millions of copies of specific DNA regions, boosting them to levels that are easy to detect and analyze. Finally, we perform short tandem repeat or STR analysis. STRs are highly variable sections of DNA that differ from person to person, making them incredibly useful for distinguishing between individuals. Together, these methods form the backbone of modern DNA analysis in the lab. When we talk about interpreting DNA profiles, the first step is comparing the DNA found at a crime scene or other evidence samples to a known reference sample, usually from a suspect or victim. This comparison helps us identify whether the profiles match, which can link an individual to the evidence. To support these comparisons, we use statistical analysis. One important tool is the random match probability, which calculates how likely it is that a DNA match could occur by chance alone. This helps us understand the strength of the evidence and its significance in a legal context. However, interpreting DNA profiles isn't always straightforward. We often encounter mixed samples containing DNA from multiple individuals or partial profiles where some data is missing. These situations require careful analysis because they add complexity and uncertainty, which can affect how the evidence is presented and understood in court. When it comes to criminal investigations, forensic DNA plays a crucial role, especially in analyzing biological evidence found at crime scenes. This analysis allows investigators to uncover vital information that might otherwise go unnoticed. One of the most significant benefits of forensic DNA is its ability to identify both victims and suspects with a remarkable level of certainty, which can be pivotal in building a strong case. Additionally, the use of DNA evidence has had a profound impact on cold cases. Many long unsolved cases have been reopened and ultimately resolved thanks to advances in DNA technology, showcasing just how powerful and transformative forensic DNA can be in the pursuit of justice. Now, let's talk about some of the challenges and limitations we face in this field. First, there are risks to sample integrity. Samples can easily become contaminated or degrade over time, which can seriously compromise the reliability of the results. Maintaining strict protocols to protect the samples is crucial. Next, the complexity of DNA mixtures presents significant difficulties. When samples contain DNA from multiple individuals, or when the amount of DNA is very low, analyzing these mixtures becomes particularly challenging. Lastly, interpretation is another area where caution is essential. Due to uncertainties inherent in the data, forensic scientists must rely on validated methods and exercise careful judgment to avoid errors. These challenges highlight why transparency and rigor are so important throughout the process. Now, let's look at some of the recent technological advancements that are transforming the field. First, rapid DNA testing has become a game changer by allowing DNA analysis to be done quickly and directly at the site. This means results can be obtained much faster, which is crucial in time-sensitive situations. Second, we have massively parallel sequencing, also known as next-generation sequencing. This technology goes far beyond traditional methods, providing a much more detailed and comprehensive view of genetic information. It's opening new doors for more accurate and in-depth genetic analysis than ever before. On this slide, we look at two impactful case studies that showcase the power of DNA in forensic science. First, a burglary case where DNA left at the crime scene led to the identification, arrest, and conviction of the perpetrator. This demonstrates how DNA evidence can directly link a suspect to a crime, providing clear and compelling proof in investigations. On the other hand, we also have a case where an innocent person was exonerated thanks to DNA testing. By excluding them as a suspect, forensic DNA helped correct a potential miscarriage of justice, proving its critical role in ensuring that only the guilty are punished. These examples highlight not only how DNA can solve crimes, but also protect the innocent. When it comes to DNA databases, privacy concerns are a major issue. Storing genetic information carries the risk that this sensitive data could be misused or accessed without proper consent. It's crucial to protect individuals' genetic privacy to maintain trust and prevent discrimination. On the legal side, there are specific standards that determine how DNA evidence is handled in the courtroom. These rules ensure that the evidence is both reliable and admissible, so it can truly serve justice without compromising fairness. Finally, the forensic community faces a challenge of balancing rapid technological advances with ethical responsibilities. While new tools offer incredible opportunities, it's essential to use them thoughtfully, respecting individuals' rights and the broader implications for society. This balance is key to responsible and effective forensic science. Now, looking ahead, we see some exciting future trends and innovations in forensic DNA analysis. First, portable DNA analyzers are becoming a reality. These devices will allow forensic teams to conduct DNA testing right at the scene, speeding up the process and providing immediate, actionable results. This field-ready capability could revolutionize how quickly investigations move forward. Secondly, advances in bioinformatics and DNA phenotyping are opening new doors. These technologies enable us to extract far more detailed information from DNA samples, such as physical characteristics or ancestral origins, which can offer deeper investigative insights. Together, these innovations promise to make forensic DNA analysis more powerful and efficient than ever before. To wrap up, forensic DNA analysis plays a crucial role in today's criminal justice system. It provides accurate identification, helping to resolve cases with a high degree of certainty. This makes it an indispensable tool for investigators and courts alike. However, the effectiveness of forensic DNA depends heavily on ongoing technological advancements and adherence to strict ethical standards. These elements are vital not only for improving accuracy, but also for maintaining public trust in the process. Balancing innovation with responsibility ensures that forensic DNA remains a powerful and trusted resource in pursuit of justice.